here are two examples, and you'll have to kind of separate these two. So we're looking first at the top, and this is a bacteriophage. This is bacteriophage lambda, and lambda is actually a phage that induces lysogeny. So in other words, it's going to actually incorporate into the bacterial genome. So here's our bacteriophage, and it is attaching to our E. coli. And notice that the rest of this stays on the outside. The capsid, everything stays on the outside. The only thing that is, for the most part, the only thing that is put into the cell is the genome. And there may be some proteins associated with that as well. One of them is going to be integrase in this case. So that enzyme also has to come in with the genome. And then if it does have integrase, if the particular virus, and in this case it has integrase, integrase is going to allow this piece of DNA, because this is a double-stranded DNA genome, it's going to allow that piece of DNA to integrate into the host genome. And what actually happens, we're going to see on the next page. So this is just the potential for this to happen. So we end up with it coming into the cell, and then it gets integrated. But at any time, one of these prophages, because now it's known as a prophage because it's integrated, at any time a prophage can be excised. So that phage DNA can be kicked out of the genome. Usually that's going to happen at a point where maybe there's an environmental change. But if it's integrated and it's not kicked out, we'll see on the next page what happens. But let's look briefly at, well, actually I'm going to change this. No, I'm not. All right, I'm going to look at HIV. So HIV is a retrovirus, and a retrovirus is an RNA virus. So it has a single-stranded RNA genome, but it carries with it an enzyme called reverse transcriptase. And reverse transcriptase is going to be really important to us when we start looking at the genomics and studying how we can manipulate DNA. So reverse transcriptase takes that copy of RNA and it makes it into DNA. And remember that in a eukaryotic cell, which we're looking at, because we see here is the nuclear membrane, here are pores, and then here's our cell membrane, and our cell membrane is where our virus is going to attach. But in order to incorporate DNA or incorporate genetic material into a eukaryotic cell, it has to be able to be made from DNA into RNA. Or excuse me, opposite. It has to be made from RNA into DNA. So it's the virus is carrying the RNA. It has to make it into DNA. And reverse transcriptase is what does that for us. And then it is also carrying integrase, and integrase is going to allow it to go into the nucleus and to incorporate in the host DNA. And remember that in this case is going to be a provirus, right? So up here it's going to be a prophage. In a eukaryotic cell, this is going to be called a provirus. Okay, and we're taking this up from the last slide. So you're seeing bacteriophage lambda here. And now lambda, first of all, an advantage that lambda has is it can be that cell, while the DNA or while the genetic material is incorporated, and it is DNA, but while that genetic material is incorporated into the genome of the bacteria, every time the bacteria reproduces, it's making more of that DNA as well. And it can stay in there for a long time until something actually kind of gives it a little oomph and says, all right, get a move on. And that's when it will come out of the genome, and it's going to start a lytic phase. And a lytic phase is where now the host genome is going to be broken down. More of all the different components are going to be made. Before, it's just living there happily and just kind of reproducing as the bacteria does. Now it's got a reason. Something has inspired it to become a virus and infect other cells. So it makes its components, including more of its genomic material, in this case the DNA, and now they're going to assemble, and when they assemble, they're now going to burst this cell and be released. So that's how a phage would work. Something like HIV is going to be a little bit different. So remember that we had our provirus here, and our provirus, just like every virus needs to make more of itself. So it's making more of the viral RNA, and the viral RNA is what's going to get packaged and sent out with each viral particle. Also, it's co-opted this machinery to make all these other parts and pieces, including the spikes that are going to go on the surface. So when this is all complete and all the parts are made, then those parts are packaged in the capsid, so genetic material plus anything it needs to take. In this case, we know it takes integrase and reverse transcriptase. So those are going to get packaged. It's going to bud out of the surface of the cell, and when it does, it's going to take that envelope and it's going to stud that envelope with these viral particles, and then it's going to move along. So it's going to break out of the surface of the cell. Now it's a virus, and now it can go and infect other cells. So latency is that inactive phase. So when a virus is integrated into the host genome, okay, remember our prophage or our provirus, but either way, it is inactive. No more virus is actually being made at that point. So it stays inactive, and it can stay inactive for an indefinite period of time. We know with HIV, we see a very long stage of it being inactive, and our body is able to keep it in control to a certain point, and then it no longer is. So the same thing happens with a bacterial virus. So latency in a bacteriophage is called lysogeny. So lysogeny is when that bacterial genome gets into, or excuse me, the viral genome gets into the bacteria, and it just stays that way. And as I said, the advantage of that for the virus is it can make more of itself and keep being reproduced with every generation of bacteria. So we have two different terms. One is a temperate phage. So now we're talking about phages again. So we're talking about bacterial viruses. So we have a temperate phage, and that has both lytic and lysogenic cycles. So our lysogenic cycles can happen until a point where usually some environmental stimulus comes along and it induces it to become lytic. And then we have a virulent phage, and a virulent phage has no ability to show any latency of any sort. Basically, it infects the bacteria, and the only thing it knows is how to break the host genome apart, make more of itself, burst the cell, and go on to infect new cells. And an example of this is separating them, and this is a little bit better than the other slide as far as showing it. But so a lytic cycle, here's our phage DNA, and it's being put into our bacterial, along with our bacterial chromosome, but notice it does not get integrated. So it stays in the cell, but it's going to start this destruction of the host chromosome. So the host chromosome is going to be destroyed, more viral particles are going to be made, more of the viral genome is going to be made, and then the cell is going to be broken. That's a lytic cycle. A lysogenic cycle is going to be where the phage inserts its genetic material, but that genetic material then is going to end up as a prophage incorporated into the host DNA, and then it's going to continue to be reproduced. And at some point, maybe it will be excised, so that prophage will be excised, it will now be separate, and it will start that lytic phase again.